Okay, students, now we will be talking about the ELISA. As I had earlier told you, that ELISA is the enzyme linked immunosorbent assay. Assay means type of testing. Okay, laboratory testing diagnosis is called as the assay. So it is enzyme linked. Enzyme linked means the here we are going to something which is enzyme link. Immunosorbent. Sorbent means something which is attaching, which is giving the attachment surface. So here once again we'll see the full form of it. It is enzyme linked immunosorbent assay. So this is a very special type of assay in which we are going to use the plate, and it is a very special plate design technique in which we are using the quantification of antigen. Even we can do the quantification of peptide, we can do the quantification of protein, we can do the quantification of hormones, and especially it is used for enzyme and antibodies. That's why it is known as the immunosorbent assay. So they are typically performed in the 96 well polystyrene plate. You all remember last lecture I told you the about the polystyrene tray. I told you that this is a very special material which gives the adsorptive surface for binding of the antigen as well as the antibodies. Yes, you all remember we had already discussed about it. So when we talk about the ELISA or this particular test, we are performing. We need to remember we are going to perform it in a very special tray. Usually, the typical ELISA tray, it is having the 96 micro wells. Students, my screen is visible to you all. Yes. Is my screen visible?
Yes, ma'am. Yes, ma'am.
Okay, we'll go ahead. Now we will discuss about the steps. So when we talk about the key steps into the ELISA, we had already discussed in the earlier lecture that first we'll talk about the principle. When we talk about the principle of the ELISA, now see antigen antibody, they make their own complex, and we have learned that antigen and antibody complex when they form, they causes the agglutination. Now, agglutination is visualized in normal test, which we had already discussed earlier. We had discussed about the Coomb test, then we have discussed about the Haemagglutination, indirect agglutination, many type of agglutination we all had discussed. In that we were just observing the clumping, but that was a very lengthy procedure, right? We all have seen that over the night we were keeping it for the incubation. So ELISA is bit faster complex, and the here best part is basic principle is we are using the enzyme. Now here we are going to use the special type of enzyme which is having capability to convert a colorless substance to a colored product, and such type of substrates are called as the chromogen. That means they are going to produce the colored. So if the color is produced, so it is very simple that yes, enzyme linked antibody is binding, it means the antigen is present. So it is a very sensitive test. Now see agglutination, we all have seen that sometimes the primary antibodies, they fail to bind, and its modification of assay was the Coombs test. Yes, you all remember in the last lecture we all had discussed about it. But now to modify, to take the antigen antibody binding to the more sensitive level, this ELISA has been designed. Now where the detection is done. Now see, we know this colored substance we always observe into the spectrophotometer. So there are very special type of spectrophotometer in which we keep this micro tray or this ELISA plate, and there we read the intensity of the color, and each well is been counted. Now we'll take a small well out of it. Now suppose imagine this is the one well out of 96 well, In that we are performing the ELISA. So first step is going to be the antigen coating. Now how we are going to coat the antigen? Say example, there is one patient A, and in that patient A we are expecting, or that person has submitted his blood sample for the detecting antigen X. Okay, now this patient A will not have only the antigen A, sorry, antigen X, right? Suppose I will just draw a little. Suppose this is a patient, okay? Example ले लेते हैं. So this patient will have antigen X, Y. He may have the Z antigen also. So many type of antigen is there. So here one only antigen of interest is shown, but what is going to be happen? I told you this polystyrene surface, it is made up of very special material. This polystyrene material allows the adherence of antigens. So antigen, only one antigen will not be there. Na? Suppose if a person is, say example, suffering from the COVID and he has submitted his blood for the COVID detection, he may also have the malarial antigen into the body. He may have the antigen for the TB. He may have antigen for XYZ diseases, right? So all these X, Y, Z all will adhere itself. X, Y. Suppose say here Z will also be attaching. Understood? So all antigen will adhere. Now, what is going to be happen along with that? Now, where these antigens are present? These antigens are present into the serum of the patient. So, we have coated this polystyrene well with the patient serum. Thereafter, we will be adding the buffer so that everything has been coated. Now after that, what is going to do, we are going to remove the buffer and we are going to wash the plate. Now this plate is adsorptive. So here you can see, now see this green ball will consider is our antigen X. So here along with XYZ everything is stuck, but here only I have shown the antigen X. So every antigen has been attached here. So now what I will do, I am going to do the blocking. Now usually this blocking is done with the blocking agent. This blocking agent are the unrelated proteins which are not going to react with the antibody molecules. It could be casein or BCA type, that is the bovine serum albumin. Bovine serum albumin is usually used. Now why it is been used? It is used to block unbound site into the well. Now in the blocking, this antigen XYZ everything is been there, so remaining thing should be, so here it, this way the blocking will be occurring, so the sides which are vacant will not be available for anything to stick, because I told you this polystyrene surface is very specific for binding of antigen as well as antibody. Now why blocking is important, student? What is going to be happen for the purpose of detection? We are going to use the antibody. So what is going to be happen? The extra antibody will start sticking here, right, if the surface is vacant. So this surface should not remain vacant, it should not remain empty, so that it is going to be a site for antibody to bind. So only one place is going to be open. This place is this your antigen's mouth, or you can say the apoptotic region of the antigen. So here only one antigen is shown for the convenience of understand, but likewise many antigens will be there, that all will be stuck over it, and remaining part will be attached, or you can say is properly covered with the blocking reagent. Now what is going to be happen? Again we are going to wash to remove the excess buffer, and after that we will do the detection. Now see, detection, if we have to find out any type of the your antigen, so we know that antigen antibody are very specific. Antibodies are designed or you can say they have the proper structure or proper binding site which attaches itself, or it's your, you can say, paratopic region to the epitopic region of the antigen. So this properly designs antibody. These are made by the special type of the cell that is called as the hybridoma cell. With the help of this hybridoma cell, special type of antibodies are designed into the laboratory. So now see what I have done. I have done the coating, I have done the blocking. Now I will be using the laboratory reagent. Now, in my laboratory reagent, I have designed the antibody against X. So, this is my X antigen. Say, this is the shape of X antigen. So, I will design the anti-X antibody, which is having the, you can say, the proper shape. So, which can bind to the X in a proper way. So now this antibody, if antigen antibody complex will be formed, only the agglutination will form, right? But I don't want agglutination. I want the proper results or proper quantitative result. I want that how much antigen is present. So for that, what I do, I will attach one type of enzyme here. Now I told you about the enzyme already, that these are the special enzyme which are attached on the Fc portion of the antibody. Now we have learned about the antibody, that antibody is having the two part. This particular region is called as the your Fc region, and this Y broad face like structure, this particular region is the Fab region, and this Fab region is responsible, or you can say it is having the site to attach itself with the antigen, and this Fc region is the effector region which on which we are going to attach the enzyme. Now about enzyme, I had already told you that these are the special enzyme which can convert the colorless substrate into the colored product. Now for that particular reason, now see, you need to remember one thing. This antibodies, they are prime, it could be the primary antibody or it could be the secondary antibody. When we are going to discuss in detail about the types of ELISA, I will tell you in detail about that. So now, for now, you just remember we are going to use the special type of antibody, and these antibodies are labeled, and you can say it is enzyme linked. So usually these antibodies, they are labeled with the alkaline phosphate or horseradish peroxidase. So these two type of enzymes are usually used. You need to remember, it is important to remember the name of enzyme. It could be asked for the MCQ purpose. So we are using ALP, that is alkaline phosphatase, or we are using horseradish peroxidase. So we are using these two type of enzyme usually, and thereafter the last stage is the detection. Now how we are going to detect it? Now see, we have followed the primary antibody treatment, wells, plate are wash. Okay, now I have added the reagent. In the reagent, enzyme linked antibodies are there. So what is going to be happen? Wherever antigen is present, antibody is going to bind. So after that, I will do the washing. Why washing is important here? Washing is very important so that the excess antibodies which are not bound, or you can say which are unbound, they will wash off. Now here, if the patient is positive, that particular time only antigen antibody complex are going to form. If the patient is negative, so that particular time all the antibodies will be washed off. Are you following? Suppose the patient is having antigen X, that time only anti X antibody can bind. Is it or not? If the patient do not have the X antigen, so there is no chance to bind this particular laboratory designed enzyme linked antibody. So all the antibodies are going to be washed off, and after that, what is going to be happen? We are going to add the substrate, and you can say the substrates are chromogen. Then thereafter, a colored complex will produce. So the choice of substrate depend on the required assay sensitivity, instrument available for signal detection. Finally, we are going to read out after catalyzation. I told you there is going to be a colored product. So visually also we can see very well a colored product will form, and this colored product will say that the patient is positive and how much antigen is present. So to see that particular thing we need to read it into the spectrophotometer. So the spectrophotometer will say that how much titer is present. So these are the basic steps of the ELISA. So now let us discuss about the types of ELISA. First type is the your direct ELISA. Direct ELISA procedure. Just now we have discussed. It was the direct ELISA only. Simply we have one antigen and we have the primary antibodies which are conjugated with the enzyme, and after that we are going to add the substrate. Now indirect ELISA, in that, now see why there is a need of indirect ELISA. Sometime what happened that these antigens they are coated on the multi-well plate and detected a very small amount. See, these are the ELISA plate and this is micropipette. We are using micropipette to fill the wells. So, student, what happened? Sometimes this intensity of color is very low. If number of antigen is very small, so that particular time a very small amount of color is been produced. So their detection is very difficult. So that particular time, what we do, we prepare the secondary antibodies over the primary antibody. Now question comes here, how and why this secondary antibody is required. Suppose antibody, anti-X antibody is going to be injected into the another type or another species animal. Suppose example, humans antibody if injected inside the mouse, so what is going to be happen, children? Yes, tell me what will happen. I will take another slide here. Suppose this is now, student, bear with my drawing. This is anti-X antibody. Okay, this is anti-X antibody. Now I have inserted this anti-X antibody into the patient. Sorry, into the another animal. Say rat. This is human antibody. Okay, this is anti-X human antibody. So if inside the mouse it is been entered, so for mouse it is what, it is the foreign substance because this antibody is not made by the mouse, right? So what is going to be happen, it is going to challenge immune system of the mouse. Now this antibody will have the various regions. Say it is having this region, so this region will be the epitope. Now this region I will make some little bit different design. Okay, this region will produce one epitope. This region will also be act as the epitope. Now we know one antigen may have the many type of epitope, right? We all have studied this. So say example, this antibody is having, now whatever design I have made, it is the epitope. Now immune system, if any antigen challenges to our immune system, so that particular time the immune system goes to the higher alert and it takes the antigen, it reads antigen in a different way. Suppose the some cells have read or made antibody against this epitope, some will produce for this epitope. Say example, that my mouth or my face is an antigen, so my ear will be one epitope, my nose will be one epitope, my lips will be one epitope, my eyes will be one epitope. Understood? So the immune system will produce the antibody against, now various part of the immune system will produce the antibody against various epitopes. So Here one antibody will be identical to it, will produce here, one antibody will be produced here. Okay, now one will be produced here, one will be produced here. So we can see that the four antibodies are been formed. Okay, now these antibodies are called as the polyclonal antibody. Now from the immune headquarter of the animal, or you can say the laboratory species, laboratory animal will be removed from its immune headquarters, that is thymus, and these antibodies will be labeled. Now point to be remember here, anti-X antibody will be normal. Okay, now it is called as the primary antibody. Now against it all the polyclonal antibody will be entered or will be used as a laboratory reagent, and this secondary antibody will be now this is primary antibody. Okay, over this now I will make only one antibody, but likewise many antibodies would be there, student. So this antibody will be enzyme linked now. Okay, so here you can see this primary antibody is there. Now these primary antibodies are not labeled. Now secondary antibody, for the convenience only one antibody has been designed, but it will be many. So now if 4 5 antibodies are attaching, so 4 5 antibodies will have the 4 5 enzyme. So 4 5 enzymes are going to convert the substrate into the product. So 4 5 times more the product is going to be formed. So this was the primary idea, and was the idea to design the more and more specific, or you can say more and more sensitive tests, so the signal strength will be more. So in indirect ELISA, what we are going to do again, we are going to prepare the antigen coated wells. Then thereafter we will what we will do, we will add the unlabeled primary antibody washing. After that what is going to be happen, antigen antibody complex will be there, but here it is not going to prepare any type of your colored product, because after that what we are going to do, we are going to add the secondary antibodies. Now these secondary antibodies are conjugated with the your enzyme, and these are going to bind with the primary antibodies, and now the signal strength is going to be more. So this was the idea of the indirect ELISA. Now students, earlier I told you that what happened, that this is, suppose say example, this is your ELISA plate. So here all the antigen will bind, X antigen will bind here, here your Y will bind, here your Z will bind, all will remain there, right? So what happens at the time of blocking, this antigen is also expressed here, this antigen is also expressed, all these antigens are expressed, but I am not interested in all that antigen. So what is done, pre-coated ELISA plate is been formed. I told you this ELISA plate can allow attachment of antigen as well as antibodies. So here these ELISA plates are made with the special type of antibodies. Now suppose I am interested to study the antigen X, right? So I will have the ELISA plate which is already bound with the anti-X antibody. So here only the speciality is there, that antibodies would be there. Okay, now see what would be the step. This is your ELISA plate, these are pre-coated ELISA plate. Okay, now here it is coated with anti-X antibody, and after that blocking is done. So that means only the mouth of antibodies are open, or you can say the variable part of the antibody is exposed and rest all part is been blocked, so nothing other than X antigen can bind over here. So this test or this enhances the sensitivity. So here if the patient is having any type of antigen, will not bound, only and only X can bind over here. And after that the procedure will be same, X will bind here, then after that we will do the washing. Then it can be detected, that antigen can be detected either the direct ELISA or indirect ELISA. In the case of the direct ELISA, what we are going to do, we are going to add the anti-X antibody again. So here the antigen is gets uh, your sandwich between. Okay, so here I have shown the indirect ELISA. So these are the captured antibody which is already coated inside the well. Understood? So this is the idea. So this first antibodies, this is already present in the well, and this type of ELISA plates for special tests like HIV, AIDS, and all is already available in the market, in which the capture antibodies are coated inside the well, and the sample containing the antigen will be added to the well. Now this second antibody will be known as the detection antibody. Okay, so this captured antigen, they are either can be detected. Now here the indirect ELISA test has been used, but it can even detect it with the direct ELISA test. So last type of ELISA is the competitive ELISA. This competitive ELISA, it includes the two step. In the first step, what is going to be happen, antibodies they are incubated with the patient serum containing the antigen. Okay, in the first step. Then second step, whatever resulting antigen antibody complex will be formed, they will be added inside the well. Now after that the coating will be done with the antigen under the investigation. After incubation period, what will happen, whatever unbound antibodies are there, that antibodies will be washed with the buffer. So more antigen in the serum sample, more and more primary antibody will be utilized, and less will be available to bind to the antigen. So what is going to be happen, amount of free antibody present here, that will be available to bind antigen coated on the well. So this amount of signal will be calculated. Lesser the color intensity developed, more antigen were present in the sample. So the main advantage of this particular type of ELISA test is that it is highly sensitive towards the compositional differences. It is present, it is giving the accurate count that how much antigen is there. So what are the procedure, that first time what is going to be happen, see these antibodies are already bound first with the serum, and thereafter the antigen antibody complex has been added, and remaining antigens will be added. You can say the patient's serum is going to be added, and thereafter it is going to be counted that how much antigen antibody complex are forming. So it is a like of competition. So for that particular reason, this type of tests are called as the competitive ELISA. So this is all about ELISA. In the case of ELISA, you all need to remember that it is the modification of the agglutination test only. And here we need to remember the type, and in type only we need to remember the basic steps. So for this note, we'll stop for now. Any doubt?